Okay, so far we've been talking about abstract, unobservable things. We've been talking about the history of our sample, but we haven't talked about anything about how you'd observe it. Right? This should give us some sense of what the shape of our trees are, but why would we care, basically, other than for theoretical interest? Well, a lot of the reason that we care is we're interested in taking sequences and trying to reconstruct what their history is and figure out what the distribution of SNPs should look like. So if this is our evolutionary tree, for instance, along each branch there's an opportunity for mutation, and this video is going to go through explicitly how we talk about that. So let's see if I can get a different color. If we have mutations occurring randomly on the tree, then this distribution of tree lengths and this distribution of tree shapes should also give us a sense of what our mutations should look like. So for instance, because this base, the deepest branch is the longest, we expect the most mutations to have occurred along that branch. And so if we think of what that means, if this mutation means that at site 734 in our sequence, there was an A to G change, so that means the ancestor had an A. Well, that means that all these individuals now will have a G at site 734, and this individual will have an A. Right, so by understanding this tree shape, we can get a sense of what our distribution of SNPs should look like. And so the first thing we can notice then is that because these shortest branches are, because the most recent branches are the shortest, right? We said that coalescences happen much more frequently down here, that we don't expect as many so-called singletons, where we'll have a mutation here that will differentiate this sample from all the other ones. So in this case, it's a little confusing because as I've drawn the tree, this one is different. It coalesces last with everything else. In a larger sample, we wouldn't expect that. The point here is that we're going to get a lot of mutations along this same deepest branch, leading to a lot of sequences. The sequences on the left half and the sequences on the right half are going to be differentiated by the largest number of SNPs.